STB1093 Microbiology Experiment 5 Assays for Specific Metabolic Activities.
According to Patel 2022, metabolic activity is the portion of metabolism that occur within an organism body. It is made up of the chemical reaction required for a living thing to survive. It has to do with how the body changes energy which is necessary for life to exist at all. Microorganisms like bacteria operate via intricate metabolic pathway directing the movement of energy and carbon within their cellular structure. This comprehension aids in differentiating bacterial species by relying on their capacity to metabolize specific substance and create unique outputs. The biochemical characteristics of bacteria, such as their food preference, waste byproduct, and enzyme generation, are critical in determining their distinct identities. Heterotrophic microorganisms display two fundamental metabolic approaches, which is fermentation and respiration. Identification of certain bacterial species hinges upon their ability to use carbohydrates for energy through either oxidative or fermentative pathway. Through the action of microbes, fermentation is a process that aids in the breakdown of big organic molecules into smaller ones. For instance, proteins are transformed to peptides or amino acids by yeast enzymes, whereas sugar and starches are turned to alcohol. Sharma et al. 2022. Also, fermentation is an anaerobic breakdown of carbohydrate, typically yield diverse end products like various acids or a combination of acid and gas, depending on the organism. Some bacteria stand out for their exclusive use of citrate as a carbon source. Detecting this trait involves inoculating bacteria onto Simon citrate agar, where the enzyme citrate breaks down citrate and convert ammonium phosphate into ammonia and ammonium hydroxide. This molecule causes the agar to become alkaline, manifesting as a distinct blue color due to the rise in pH, thereby indicating the bacterial species' unique metabolic capabilities. The specific carbohydrates were added in 1 ml quantities to the purple broth or fennel red broth tube, which already contained a protein source to support bacterial growth and a pH indicator. The tubes were labeled accordingly. The next step in this experiment was a set of carbohydrate supplemented tubes was inoculated with each of the test bacteria. The tubes were then incubated at 37 degrees Celsius for 24 to 48 hours. Next, after the incubation period, the color of the tube was observed. Acid production was indicated by a change to yellow. The Durham tubes were examined and partial of complete filling was indicative of positive gas production. Lastly, the results were recorded. In this experiment, the test cultures were carefully introduced into tubes filled with a specific type of broth called MRBP broth. Each tube was meticulously labeled to ensure tracking of the bacterial cultures added to the various tubes of the specialized broth. Subsequently, the inoculated tubes were then incubated at 37 degrees Celsius for 24 hours to 48 hours. After the completion of this incubation time, 1 ml of the bacterial culture from each tube was cautiously transferred into a new sterile test tube. To this portion of the culture, 0.5 ml of baric reagent A and 0.5 ml of baric reagent B were added. Next, the mixture was vigorously shaken for 30 seconds. The tubes were observed for the gradual formation of a pink to red color, indicating a positive test for acetine. Some cultures reacted within 1 minute, while others took 1 to 2 hours. The result was recorded. Broth left over from the cultures grown for the Voges Proskauer test could have been used for the Meti-Red test. If the Voges Proskauer test was not performed, tubes of MRBP medium were inoculated with each of the cultures and incubated at 37 degrees Celsius for 24 hours to 48 hours. Meti-Red solution was added to the cultures in a few drops. An immediate red reaction was indicative of mixed acid fermentation, while a yellow reaction indicated a negative test. The results were recorded. The slants of Simon citrate agar were labeled accordingly. The slants of Simon citrate agar were inoculated with each of the test microorganisms, namely bacteria A and bacteria B. The tubes were incubated at 37 degrees Celsius for a duration of 24 hours to 48 hours. Following the incubation period, the color of the tubes were observed. A royal blue color were considered positive test, while a green color indicate a negative result for citrate utilizations. The result were recorded accordingly.
Let's look at the result obtained from the test A, which is acid and gas production from carbohydrate fermentation. In both E. coli and Staphylococcus aureus, carbohydrate is present. However, acid production and gas production only occurred in E. coli. Therefore, only E. coli gave a positive reaction in this test, as we can observe in the change of color of both the solutions. Looking at the result of the next test, which is Voges prokaryote test, E. coli gave a negative reaction, whereas Staphylococcus aureus gave a positive reaction. There is a change in color from yellow to red in the solution containing Staphylococcus aureus, whereas no change in color in E. coli containing media. The next test is methyl red test, in which both the E. coli and Staphylococcus aureus gave a positive reaction. There is a change in color from yellow to red in both the solutions. The last test is citrate utilization test. E. coli gave a negative reaction in this test, while Staphylococcus aureus gave a positive reaction in this test. This can be observed from the change of color of the agar from green to blue. Let's look at the discussion of test A. There is a difference in color between solution containing E. coli and S. aureus. The solution in test tube A turns from red to reddish yellow, while test tube B turns from red to bright yellow. There were also bubbles of gases found at the bottom of test tube B. Referring to Hartlin 2022, in the event if there is a bubble appeared in the Durham tube, fermentation has taken place. Referring to the fact, the solution containing E. coli gives positive result, while solution containing S. aureus gives the negative result. However, as we look at the result that we observe, as we can look at the picture beside, there is no clear space formed in the Durham tube. This may be due to the incomplete fermentation in the bacteria or insufficient time. Let's discuss test B, Voges prokaryote test. When 0.5 ml of barium reagent A and B is added into both of the test tubes, there is a gradual formation of red color in test tube A containing Staphylococcus aureus and a slightest change in test tube B containing E. coli, where the bright yellow turns to deep yellow solution but not red. This is because the butanediol pathway is carried out by S. aureus but not by E. coli. This is an appropriate reaction to distinguish between bacteria. Staphylococcus aureus able to undergo this butanediol pathway as it contains enzymes and genetic machinery needed to carry out fermentation pathway, which enables the S. aureus to convert glucose into acetone, while E. coli lacks the entire set of enzymes needed for this pathway. Methyl red test. Both E. coli and S. aureus containing media gave a positive result in this test. When methyl red indicator is added, there is an immediate change in color in both test tubes. This is because E. coli and S. aureus produces a range of end products during mixed acid fermentation, such as carbon dioxide, ethanol, acetate, and formate. This causes the medium's pH to lower it and causes the methyl red to detect the pH change and turns the solution to red, indicating the solution is acidic. However, this is not a suitable test to differentiate between these both bacteria, as the both bacteria gave the positive result. Growth of microorganisms on this medium results in a rise in the pH that causes the pH indicator to turn from green to royal blue. Referring to the observation that is obtained, the agar labeled A gives positive result in this experiment as it turns blue and the growth of bacteria is visible on the slant agar. Therefore, the bacteria present in this agar is S. aureus. However, there is no trace of bacterial growth or color change observed in agar B. Therefore, it is E. coli bacteria. According to Subquota 2020, when the medium's pH rises above 7.6, the alkaline carbonates and bicarbonates that are created as byproducts of citrate causes the green to turn blue. In addition, due to its facultative nature, coli usually uses glucose as its preferred carbon source. Although E. coli is capable of breaking down a wide range of sugars and other substances, it still lacks the enzymes such as citrate lyase, and they use citrate as a carbon source. Now let's answer the questions. Question 1. Your microorganism grows in the tube with glucose as shown by an increase in turbidity, but the tube does not turn yellow. Why? A rise in turbidity signifies the growth of microorganism undergoing fermentation without the production of gas or acid. As a result, the medium's increasing turbidity indicating rising microbial mass. Question number 2. What gas other than carbon dioxide might be produced and collected in the Durham tube? Other than carbon dioxide, hydrogen gas also can be discovered in the Durham tube. Next, how could you detect acid and gas production using a solid medium? It can be done by using nutrient agar, as it can act as a medium for microbial growth. Next, questions on test B. If the Voges prokaryote test is run too early or too late, it will be negative even if the microorganism is carrying out the butanediol fermentation. Why? Acetone is an intermediate in the butanediol fermentation pathway and is eventually converted to 2,3-butanediol. There might not be enough substrate for the BP reagents to react with if the test is carried out too soon before enough acetone has been produced. Besides, acetone will further metabolize into 2,3-butanediol if the fermentation process is started too late, leaving insufficient acetone for the BP test to detect. These both conditions will lead to negative results. Next question, why is the Voges prokaryote test important in microbial taxonomy? It is used to determine if a microorganism produces acetylmethylcarbonate from glucose fermentation or not, which enable to distinguish between microorganisms. Metabolic test. Question 1. Can a microorganism carry out both the butanediol and mixed acid fermentations? Indeed, microbes are able to carry out both the mixed acid fermentation as well as the butanediol fermentation pathway. Question number 2. Can a microorganism be both methyl red and BP positive? Explain. Yes, it is possible for a microorganism to be both MR and BP positive. Microorganisms that experience sequential metabolic shifts during growth frequently experience this situation. While BP detects the production of acetone, MR detects the production of stable acids like lactic acid and acetic acid that lower the pH of the medium. This results in a positive MR test. A positive BP test could also result from this. Citrate utilization. Question 1. Why does the pH of semen citrate agar increase if citrate is utilized? Alkalinity is increased when the bacteria convert the ammonium salts to ammonia through their metabolism of citrate. When the medium's pH rises above 7.6, the bromothymol blue indicator in it turns blue instead of green. Question number 2. The microorganism grows on semen citrate agar. The slanted portion is blue, but the bud remains green. Explain. The color change in the slant but not in the bud may indicate that the microorganism is utilizing citrate, but the extent of utilization may vary between the slanted and bud portion. On the slant, where the agar surface is exposed to air, the citrate utilization process mainly takes place. The aerobic conditions on the slant may encourage the bacteria to use the citrate more effectively, which raise the pH and cause the color to change from green to blue. But because the tube's bud is more anaerobic, citrate utilization there may be minimal or non-existent, which would explain the lack of color change. Question number 3. The IMB tests are a series of 4 tests that are useful in identifying gram-negative enteric bacteria. How would the IMBIC test differentiate E. coli and Enterobacter aerogenes? Based on their unique metabolic traits, E. coli and Enterobacter aerogenes can be distinguished. In indole production, E. coli is typically positive, whereas Enterobacter aerogen is usually negative. This is due to the presence of tryptophanase enzyme in E. coli. Next, in methyl red test, E. coli is typically positive, exhibiting a stable acidic end product from glucose fermentation, whereas Enterobacter aerogen is often negative. Then, in BP test, E. coli is usually negative, while Enterobacter aerogen is positive, indicating the production of acetone. Lastly, in citrate utilization test, E. coli is negative for citrate utilization as it does not grow on semen citrate agar, and Enterobacter aerogen is positive for citrate utilization, leading to the growth on semen citrate agar.